Chapter 3.1, Cell Theory. Key concept: cells are the basic unit of life. Cytology is the study of cells. The prefix cyto means having to do with cells. The suffix ology means the study of. Cytopathology is the study of cellular disease and the use of cellular changes for the diagnosis of disease. Cell biology, which is what we're doing right now, is the study of normal cellular anatomy, function, and chemistry. Cell history. Robert Hooke is credited for, in 1665, being an English scientist who also looked at lots of things surrounding him. Remember, this was a time before they had Instagram and TikTok. And so scientists at the time were often hobbyists. They had other jobs, but they would observe things in nature around them. Robert Hooke was especially interested in looking through a microscope. He looked at a thin slice of cork, which is just dead cells from an oak tree, through a compound microscope. What he saw, he drew. You can see right here, this is his drawing. He drew, and they observed tiny, hollow, room-like structures. He called these structures cells because they reminded him of the rooms that the monks lived in. He only saw the cell walls because the cork cells weren't alive. Anton van Leeuwenhoek, roughly the same time as Hooke, was a Dutch fabric merchant and amateur scientist. He liked to look at blood, rainwater, and scrapings from teeth, gross, I know, through a simple microscope that he developed. It looks a little bit like a can opener over here. He observed living cells, and he called them animalcules because they look like little tiny animals. Some of those animalcules that he observed we now call bacteria. Anton van Leeuwenhoek is known as the father of microbiology. The co-founders of cell theory, Matthias Schleiden and Theodor Schwann, they were both German. Matthias Schleiden was a botanist, meaning he studied plants. So obviously he viewed plant parts under a microscope. And he is the one that discovered and made the statement that plants are made up of cells. Theodor Schwann was a zoologist, meaning he studied animals. He viewed animal parts under microscopes, and he discovered and made the statement that animal parts are made of cells. Then Rudolf Virchow came along in 1855, another German. He was a physician and a politician, and his platform, or what he ran on, was for the advancement of public health, so telling people it's not okay to throw your human waste in the streets. This is 1855, remember, this is pre-Civil War era in this country. He was the first one to recognize what we know of as leukemia cells now. He stated that all living cells had to have come from other living cells. There was no such thing as spontaneous generation. Cells didn't just magically appear out of thin air. He was an important contributor to the cell theory. Cell theory itself grew out of the work of many scientists and improvements in the microscope. Many scientists contributed to this cell theory. There's our pictures of Hooke, Leeuwenhoek, Schleiden, Schwann, and Virchow. Those are the big five that we look at. They are the ones that we will make the foldable in class about to help you remember what their contributions were. More was learned about cells as microscopes improved. The cell theory, we keep talking about it, and all it is just a unifying concept in biology. The cell theory has three principles. The first is that all organisms are made of cells. Here we can see some skin cells. They look kind of funny, but their structure determines their function, which is to protect. The second part of cell theory, all existing cells are produced by other living cells. So they have to come from other cells. And finally, the cell is the most basic unit of life. If you get smaller than a cell, it's no longer considered living. The two types of cells that we're going to talk about are prokaryotes and eukaryotes. A prokaryotic cell lacks a nucleus and most of the internal structures of the eukaryotic cells. So prokaryotes are very basic. All cells, however, share certain characteristics, like the fact that they're microscopic. One of the largest single cells is eukaryotic, and it's a human egg cell. A human egg cell is big enough to be seen with a naked eye. It's roughly the size of a period at the end of a sentence in your textbook. All cells are enclosed by a membrane. That phospholipid membrane we'll look at later on. And all cells are filled with cytoplasm, which is just the jelly gooey-like stuff that's inside. So if we compare them, the types of cells are prokaryotes versus our eukaryotes. Prokaryotes are simple cells. Eukaryotes are more complex. Prokaryotes don't have a nucleus, and they don't have membrane-bound organelles. Eukaryotes, however, have membrane-bound organelles and a nucleus, sometimes more than one. Prokaryotes have circular DNA and ribosomes. Eukaryotes possess more complex DNA in the shape of chromosomes. Examples of prokaryotes are like bacteria and archaea. Those are the two domains of prokaryotes. Examples of eukaryotes are plant and animal cells. Eukaryotic cells that have a nucleus, sometimes they can have multiple, but this example right here you can see it is stained purple, and you can see those little orange things that are in there. Those are the mitochondria, and that's going to be in chapter 4 when we talk about cellular energy. That's where cellular respiration occurs. Prokaryotic cells do not have membrane-bound organelles. They're very basic, very simple. You can also see the difference in the size. The prokaryotes are much smaller. They both have cell membranes, they both have cytoplasm, and they both have some form of DNA.